Hello everyone, my name is Zhou from Tianjin Medical University, which is located in the north of China. First, I would like to thank the organizer for invitation. My talk is about myeloid-derived suppressor cells and infant immunity. So it has been demonstrated that early life, including the embryonic stage and neonatal stage, is very important for the health of whole life. So in the pregnant, during pregnancy, if the pregnant mother were exposed to some bad factors such as pathogen infections, stress, or the disruption of the circadian rhythms, so it can affect the development of the immune system and cause a long-term effect, which can enhance the susceptibility of adult to immune disorders. So that's why it's very important to study how the immune system was regulated during early life. Compared with adults, the infant, their immune system are relatively immunotolerant. So this immunotolerance is important for the to allow the gut microbiota colonization, which usually happen within several days after birth. So if the infant cannot tolerate the colonized microbiota, that can cause inflammation in intestine, such as necrotizing enterocolitis or NEC. NEC is one of the common emergencies, which usually happens with preterm infants or very low body weight infants. So the incidence of AEC in very low weight infants is around 7 to 11%, while its mortality is pretty high. It's like 13% of yearly hair. So the pathogenesis of AEC is generally caused by the immature immune system of the preterm infants, so they cannot tolerate the colonized microbiota and cause hyperinflammation of the intestine. And so for the NEC, they cannot be treated properly, so it can develop into sepsis and death in short term. And mm, Previously, we found that we took a spleen from the mice with different ages, and we found that for the neonatal mice, so their spleen actually has this clear expansion of myeloid cells compared with adult control. So next, we want to know the function of these expanded myeloid cells. We took the myeloid cells from neonatal mice and co-cultured with T cells. And we found that these neonatal myeloid cells display very strong immunosuppressive activity. That means they are immunosuppressive myeloid cells. Um, you know that immunosuppressive myeloid cells were termed myeloid-derived suppressor cells. Usually it happens with tumor or pathogens or other pathological conditions. So under tumor or infections, so they secrete the soluble factors and that can cause the generation of MDSC in bone marrow. After generation of in bone marrow, MDSC can migrate to the peripheral tissues and suppress the function of effector immune cells such as T cells and NK cells, and also can cause the generation of regular T cells. So the overall effect is MDSC can cause immunotolerance against the tumor or the pathogens, which actually can promote the disease. So MDSC was considered as a key player in the immunotolerance under pathological conditions. After we found that MDSC can actual appearance of MDSC in neonatal mice, we actually also look at the mice during lactation. Actually, we did not find the MDSC expansion in mice during lactation. That means this phenomenon is specific for neonatal mice. And later on, we took up we collected samples from the blood of the clinical infants, and we found that for clinical infants, actually, they also contain much higher level of MDSC compared with adult control. So that means this phenomenon was observed in both mice and humans. So next, we want to ask what drives the chance appearance of newborn MDSC. And in order to answer this question, we perform transcriptional analysis by R-seq. We took up we sought the myeloid cells from the neonatal mice and as well as the adult true mice. And we found that one of the genes actually was very interested, and we lactoferrin was upregulated in neonatal MDSC, and the lactoferrin protein actually was one of the abundant proteins in breast milk, as well as in some formula they also contains lactoferrin, and it has been demonstrated that the development of immune system in neonates actually was largely affected by the nutrients provided by their mother. So that's why we pick up lactoferrin for the study. And first, we treat the bone marrow cells with lactoferrin. We can see that lactoferrin can increase the cell number of the MDSC, and also it can enhance their immunosuppressive function. That means lactoferrin could induce the generation of MDSC. And next, we perform cross-fostering experiment. We use the lactoferrin Q mice as mother. That means the breast milk from the Q mice does not contain lactoferrin protein. So if the baby mice were fostered by the lactoferrin-2 mother, so they cannot generate enough number of MDSC. That means breast milk-derived lactoferrin could promote the generation of MDSC. And to make the story shorter, actually we found that the lactoferrin protein even secreted from the neonatal myeloid cells or from the breast milk. So they can bind with the receptor LRP2 located on the membrane of the myeloid cells and cause the generation of the immunosuppressive factors in myeloid cells, and which can render myeloid cells acquire immunosuppressive function and suppress the T cell proliferation and contribute to the immune tolerance in neonates. So next, we want to know the significance of MDSC in neonates. So we set up the AEC model. So you can see that after induction of AEC in neonatal mice, so there are clear intestinal inflammation and necrosis. And next, we modulate the level of MDSC in the AEC mice, either MDSC transfer or MDSC depletion. So we can see that if we provide extra MDSC, so it can increase the survival of the AEC mice and reduce the intestinal inflammation. Well, if we deplete the MDSC using antibodies, and it can lower the survival rate and increase the intestinal inflammation. That means the presence of MDSC could protect the mice against the inflammation. And we mentioned that lactoferrin can promote the generation of MDSC. So if we feed the newborn mice with lactoferrin, actually it can increase the survival rate and reduce the intestinal inflammation and display a similar protective effect. And next, we collect the blood sample from the clinical infants, and we try to see how about the MDSC and the function. And flow cytometry analysis showed that for the AEC patient as well as the preterm infant, so they contain a much lower level of MDSC compared with term control. So, and also the concentration of lactoferrin protein was positively correlated with the number of MDSC in infants. And so next we want to know what's a potential therapeutic value of MDSC in neonatal inflammation. So we took a bone marrow from newborn mice and culture in the presence of lactoferrin protein for 2 days to induce the generation of MDSC. So after 2 days culture, we can see that the number of the MDSC was increased, and also actually the immunosuppressive activity was enhanced. that means their MDSC. So next we transfer the lactobacillus-induced MDSC to the AEC mice. So results show that lactobacillus-derived MDSC they display a much stronger protective effect compared with control MDSC, which included the HE staining of the intestine as well as the survival rate of the AEC mice. So a brief summary. So first, we found that there is a transient appearance of immunosuppressive metal cells, MDSC bodies, and the breast milk derived protein like fur could drive the appearance of newborn MDSC. And the presence of MDSC could protect the newborns against inflammatory disease such as AEC, and AEC patient and preterm infants, they display lower level of MDSC with impaired function, and lactobacillus-derived MDSC may have therapeutic value in AEC. So actually, this is just a cartoon to show the potential value of MDSC in neonatal inflammation. So here is a full-term neonates, and here is a preterm neonates. So compared with full-term neonates, preterm neonates, they have a lower number of MDSC as well as lower concentration of lactobacillus protein. So based on this defect, so we can use lactobacillus to induce a generation of MDSC and then adopt transfer into the AEC patient, which may have therapeutic values. So next, I would like share you with some unpublished study about the neonatal MDSC, and it has been demonstrated that the disruption of the motor rhythm during pregnancy can increase the incidence of preterm birth. And so we based on this knowledge, so we ask whether motor rhythm could affect the susceptibility of neonatal inflammation. Actually, this has never been studied yet. So in order to answer the question, we set up the circadian rhythm disruption model in the pregnant mice. So for the control is 12 hours day and 12 hours night. For the CRT model, in every other day, we put 4 hours in advance during the night. So after the baby mice were delivered, so we set up the ADC model. So results show that if the mother actually was subjected to the CRD model, so the pups they delivered, they have lower survival rate after AEC induction. And so also for the inflammation, intestinal inflammation, so they have higher intestinal inflammation compared with control. That means the disruption of the motor rhythm aggravate the AEC severity. So in parallel, we found that if the mother was subject to CRD, so the neonatal MDSC they have diminished the function of neonatal MDSC, and next if we transfer MDSC to the CRD delivered pups, so they can rescue the AEC severity. That means that MDSC mediated the effect of the circadian rhythm on the AEC severity. So next, we performed cross-foster experiment. So we show that so if the control mice were fostered by the CRT mothers, so they will have a lower survival rate of AEC. While the other hand, so if the CRT pups were fostered by the control mother, so they have a better survival rate. That means the effect of motor rhythm on AEC is post-natal determined. So in parallel, we found that breast milk derived factor may affect MDSC in the CRT model. And next, we ask which factor was affected, which nutrients were affected by the CRT model. So we perform on-target metabolic screening from the blood as well as breast milk. So consistently, actually, we found that DHA actually was reduced in blood from the pregnant mice as well as the neonatal mice, as well as in the breast milk of the CRD group. So that's why we pick up the DHA for the study. So DHA actually is a very important nutrient in the breast milk. So it has broad immunoregulator effect such as anti-inflammatory effect, and also DHA can improve the brain development in the neonates. So first we show that the CRD, the pregnant mice, if they are subject to CRD, so the expression of DHA synthesis enzymes was downregulated in their liver. So that means they cannot efficiently synthesize DHA. So next we provide extra DHA to the CRD mice. So from embryonic state until the pups were delivered. So after DHA supplementation, so we found that the neonatal MDSC, so their function was enhanced, so they can efficiently suppress T cell proliferation. And also, actually, we provide PAPs from the CRD mice, CRD group, so DHA can still can enhance the MDSC function. And next, we'll ask how DHA regulate the neonatal MDSC. So we performed the RNA-seq and as well as functional study. We found that the mitochondrial function of MDSC was reduced in the CRD group, the CRD delivered PAPs. So including the reduced mitochondrial mass as well as lower mitochondrial membrane potential. So we confirmed the mitochondrial function dysfunction using Seahawks assay. And in the other, actually, meanwhile, if we enhance the mitochondrial function using small molecules outbreeds, so we can see that if we enhance the mitochondrial function, that can rescue the MDSC, they can rescue the MDSC function from the CRD delivered paths. Lastly, actually, if we supplement the DHA to the CRD mice and until the capsule delivered, so we can see that the DHA can rescue the NEC severity from the CRD group. That means the DHA supplementation could relieve NEC severity. So even we provide the DHA after the mice were delivered post-natal DHA supplementation, so we can observe the protective effect of DHA on the AEC severity. So in summary, actually, we show that circadianism under healthy conditions, so the pregnant mother actually can produce DHA, consistent DHA, which actually can transfer to the pups through breast milk. And the DHA can promote the generation of neonatal MDSC, which can contribute to the tissue homeostasis. Well, if the mother was subject to the circadian rhythm disruption, so they have a lower level of DHA, which actually can cause dysfunction of the neonatal MDSC and contribute to the tissue inflammation such as NEC. So here we actually demonstrated that MDSC actually has very important physiological functions in early life, including the protection the infant against some inflammatory disease. So I would like to thank the team members, Cui Zhaohai, He Yumei, and Liu Yufeng, and also thanks the collaborator, Dr. Dimitri Gabrilovich, and also the clinical samples from different hospitals. And this study was funded by the National Center Foundation of China, as well as the high-level talents of Tianjin Medical University. Thank you.